In this video, we'll start our conversation on organic compounds. And organic compounds, we'll talk a bit about why carbon is especially well suited for biological organisms. We'll talk about functional groups. We'll talk about polymers. Talk about the functions of carbohydrates, lipids, and proteins. That will probably be in a subsequent video. And eventually we'll talk about enzymes, function, and the structure and production and function of ATP. And also talk about other nucleotides and their functions. So the next series of videos really will cover the organic compounds. But in this one, we're going to focus in on largely carbohydrates. But before we get going, let's talk a bit about organic chemistry. It's the study of components containing carbon. That's one definition, and it's workable for us. Now, there are four categories that everything falls into. You're either carbohydrate, a lipid, a protein, or a nucleic acid. And in our video, we'll be looking at basically carbohydrates in this current video. So let's start talking a bit about carbon, because of course carbon is one of the most common elements we have, and it's a beautiful element in that it allows itself to be bound to so many other atoms. And the reason is that carbon has 4 valence electrons, and remember, in your outer shell you'd like to have 8, so this gives a lot of flexibility. So it binds with other atoms that can provide 4 more electrons to help fill the valence shell. And the carbon atoms can actually bind with each other also, and they form something that we'll call, and I'm going to change color here, just highlight it, the carbon backbone. Now, what do we mean by this? Is that carbon can bind onto itself quite easily, and of course it'll bind onto other things. So we can end up with these long structures, which are the carbon backbones. Not only can it form chains, it can also form rings. It can actually fold in on itself this way. Those are little carbons, and it can bind in such a manner that it can actually have rings. And it's so adaptable that you can then have side chains coming off those rings, and those chains could have their own side chains. So you can end up with quite complex molecules because of the carbon backbones. Now, we're going to talk a bit about functional groups. Functional groups, think of them as like mini apps that you can plug into these structures, these molecules. And they're small clusters of atoms that you attach onto the carbon backbone. And these functional groups really kind of give the flavor as to what type of molecule this is, and gives a pretty good clue as to what it will do. So it determines many of the properties of the organic molecules. And we're going to see the major ones we're going to look at are the hydroxyl group, the methyl, the carboxyl, and the amino, and the phosphate groups. Now these groups you have to know. And in our textbook, there is a fabulous diagram that summarizes everything, but we're going to walk through it quickly. Make sure you go back, spend some time to look at it. First one we're going to look at is the hydroxyl, and let's zoom in on this. So when we talk about hydroxyl, we're talking about an OH. This is like a little plug-in of OH, and you can see the structure right there. Typically it occurs in sugars and alcohols. They tend to have a lot of this OH. The second group that we're going to look at is the methyl group. And a methyl group has a carbon. So we have a carbon, and we have 3 hydrogens attached to us. So it's CH3. Very common in fats, oils, steroids, amino acids. The next group is the carboxyl group, and that's represented by a C, 2 O's, and an H. And you can see it here. Here's a carbon attached to oxygens. You notice in this case it's a double bond to this oxygen, and then a bond here to this oxygen, and here we have a hydrogen right there. So that gives us the carboxyl group, which is COOH. And amino acids, sugars, and proteins is where we tend to see this. The next group would be the amino group. The amino group, and you notice that what shows up here, nitrogen shows up. It's a very unique and really distinguishes it. So we have the amino group, which is NH2. We can see it here, the N, two hydrogens, and bound onto this would be a carbon. So really these functional groups, what they do is they plug into carbon structures, carbon backbones, carbon rings, etc. The last group we're going to look at is the phosphate, and of course with a name like that, not surprisingly, look what's in the middle. We have basically the phosphorus right there. We have 1, 2, 3, 4 oxygens, and we have 2 hydrogens, and there's the carbon that it plugs into. So this whole thing is the unit right there. That's the functional group. So that's phosphate, H2PO4. Nucleic acids and ATP is where we really see these. Now, as I said, this is something you have to know. It's so basic. It's fundamental to any discussion of the molecules in biological systems. So take time, invest it, go back, learn this table, and become very comfortable with the hydroxyl, methyl, carboxyl, amino, and phosphate groups. Now we'll just say a little bit about what monomers are, monomers and polymers. Now macromolecules, macro means large. They're very large organic molecules, and because they're large, they tend to have a very large molecular weight. Poly means many. So polymers are macromolecules made of repetitive series of identical or similar subunits, and we'll call those monomers. Right, so we'll talk a bit more about what a monomer is, such as starch. Starch is a polymer made of about 3,000 glucose molecules or monomers. Polymerization is the actual process of joining the monomers to form a polymer. So that's the actual process of sticking these individual subunits together to make the larger molecule. Now there's 2 ways this can happen. What I'm going to do is I'm actually going to skip this slide. You should have a look at it, but what I'm going to do is look at the next slide, which is the diagrammatic representation. I'll spend time, but make sure you go back and look at this slide in your notes, because it just gives the but I'll walk through it. What we have here are 2 processes. Dehydration synthesis. Look at the name, break it down. Dehydration is removal or reduction in water, right? Synthesis is building up. The other process is hydrolysis or hydrolysis, often people say. Hydro for water, lysis for snipping or breaking apart. So we have these 2 processes, and we're going to look at it. Here's one, and there's the other process. So let's start with the dehydration synthesis, which is basically this process right up here. What I do is I'm going to take 2 monomers. This could be 2 sugars. Now, here's a little hydroxyl group sticking out. So there's 1 sugar. Here's a second sugar. And what happens is the 2 hydroxyl groups come close to each other. They're oriented close to each other. And what I do is I pull off a hydrogen from one, and I pull off an OH from another. So they've now been removed. Now, of course, if I have a hydrogen that's being liberated and oxygen, they want to go on and they'll form a water molecule. Now that I've pulled off the hydrogen and the OH group, this allows basically this oxygen to bond straight to this molecule, and we can see that. So what have I done? I've now chemically bonded these 2 via the oxygen. I removed a hydrogen and an OH group, and that functionally gave me a water, and that's where the name dehydration, because I pulled what's equivalent of a water molecule out, and it resulted in synthesis or a buildup. The next one is hydrolysis, and it's basically the reverse. So if you know one, all you have to do is reverse the reaction for the other. And so we're going to look at hydrolysis. In this case, I already have these, say, sugars bonded together. Now I want to break them apart so I can use each sugar independently. And in this case, I'm starting with the oxygen, the bond there with the oxygen. So I'm going to come along and I'm going to basically break the bond. And in order to break the bond so it doesn't reform, etc., I'm going to take a water molecule and I'm going to have a hydrogen from it. I'm going to have an OH from it. I'm going to use both of those to basically repopulate the functional groups here. So I've gone from what was a dimer. Dimer means basically two monomers put together. I'm going to break the bond, but I'm going to need the equivalent to one water molecule. So one hydrogen, one hydroxyl, they go and reattach, and now both of these are separate molecules, so I can use them, say, in a metabolic process. So those are the two reactions to look for carbohydrates. It's dehydration, synthesis, and hydrolysis, or hydrolysis, people often say. When I'm looking at carbohydrates, first of all, they are a hydrophilic organic molecule. Some examples are the sugars, the starches. They have a very typical sort of ratio of carbon to other things. So N means number. That's the number of carbon atoms. And usually what I have is this ratio of hydrogen to oxygen associated with it too. We're going to see some examples as we go through. Now, a couple of things that make you looking at a carbohydrate, sugar especially, is OS, like ribose, glucose, maltose, galactose, fructose. They all end in OS. And saccharose also gives you an indication you're probably looking at a sugar. Monosaccharides, disaccharides, polysaccharides. So here's some little cheat sheets, bits of words that are helpful to know. Now, there are three important monosaccharides. Remember, saccharides for sugar, mono for one. That's glucose, galactose, and fructose. They all have the same molecular formula. They're all isomers of each other, but of course, we know that isomers can have very different, basically, usabilities. So, produced by digestion of complex carbohydrates. Glucose is, say, our blood sugar. Let's have a quick look at three of the major monosaccharides. I would not expect you to memorize the actual structure of these and be able to write them out, but let's have a quick look. You notice here's glucose. Let's zoom in a bit on it. You see here's glucose, and you can see that, by the way, wherever they have a little bend here, assume there's a carbon there. They're just doing they're cheating. If there's not a carbon there, then they'll tell you there's no carbon there, like that. That's sort of the nomenclature here. So assume that there's a carbon at that angle, and except if there isn't, then they'll tell you what it is. And of course, attached to the carbon, not surprising, we have hydrogen, and look what's showing up. Hydroxyl groups, right? We have hydroxyl groups all over the map. Very typical. Now this sugar happens to be 1, 2, 3, 4, 5, 6-sided. So it's one of the 6-sided sugars, and that's glucose. Have a look at galactose. So it looks very similar. The only difference, if you go through it, the difference that you'll notice right here is here, right? Some of the orientation of the molecules in the 3 dimensions of it. So that's galactose. Once again, it's a 6-sided sugar. And if I look at fructose, you can see this one's interesting. This is a 5-sided sugar. Now, as I said, I would not ask you to replicate these molecules in terms of their 3-dimensional structure in a test or anything like that, but I think it's important they get introduced and you get a visual image as to what these look like. Now, a disaccharide, di means 2. Disaccharides are a sugar made of 2 monosaccharides, and some examples of that would be things like sucrose, sucrose, lactose, and maltose. Now, that being said, What are they made of? Now, I'm not going to hold, once again, I'm not going to hold you responsible for the components of it, but let's quickly introduce them. That a sucrose is basically a glucose and a fructose, whereas a lactose is a glucose and a galactose. So those are two different monomers that have combined to become the disaccharide. And maltose is a glucose plus a glucose. So all these are sugars because they end in O's, but these disaccharides have different combinations of the monosaccharide sugars. And you can just have a quick look. I'm not going to walk through it in great detail, but certainly take a few minutes and just sort of get a visual overview as to what these look like. You can see that, there we have the typical bonding, right? That type of bonding is as a result of dehydration synthesis. And go back and look if you don't quite recognize it. Go back a few slides. Now, carbohydrates, we can also consider them oligosaccharides. What does oligo mean? Oligo means few. Okay, it's not a million of them. It's somewhere, it's kind of between, hardly any and a whole bunch. I know that's not very, very scientific-y, but almost every book has a slightly different definition as to what is an oligosaccharide. So it's sugars made of more than a handful of, or I should say, large molecules made more than a handful of independent monomer sugars, but we're not talking thousands of them linked together. Now, a polysaccharide is when we do start to link together long chains of monosaccharides. Here our book says at least 50, some say at least 100, but give you an idea. And there's three big ones. There is glycogen. That's the only one that we actually have that we make as humans, and we tend to find it stored in things like the liver, muscle, brain, the uterus, and the vagina. And they're stored there for various reasons, and we'll leave that discussion for later. Starches are energy storage in plants. Now, luckily, we do have the enzymes to break the starches apart. So we can take plant materials and we can break up their polysaccharides and get the individual sugars and use it for our own metabolism. Now, there is cellulose, which is a structural monomer in plants. And now, unfortunately, we don't have the enzymes to break the bonds of this structure. So it can't be broken down, but it does form an important role in humans, is that this can contribute to human dietary fiber because it's indigestible to us. So just a reminder, we have glycogen, which we can make and we can use, starch, which is made by plants, but we can use it because we have the enzymes, and then there's cellulose, which is made by plants, but we do not have the enzymes, so we cannot use it, and it really functions as basically a dietary fiber. Here's just an example of glycogen. You can see these are all individual. These are all individual sugars bonded together. You can see the bonds that occur there, and of course, if I want to liberate them, I have to come along and break the bonds through hydrolysis, and then I could have an individual sugar in order to use. It's a good storage mechanism. Now, in the world of carbohydrates, often what we do is we start to add things to the carbohydrates. Now, what's the other advantage about carbohydrates? Carbohydrates are quickly mobilized as an energy source. And really what we like to do is take carbohydrates and get them down to the glucose level to use them. And they have a big role in the creation of ATP, adenosine triphosphate. Now, often what happens is carbohydrates will hook up with a different type and combine with a different type of molecule. And we call these conjugated carbohydrates. So it means they're covalently bonded to either lipid or, say, a protein. And here's some names. So not surprisingly, a glycolipid, which would be a sugar and a lipid combined. One of the places we see this is on the external surface of cell membranes. Then we also have excuse me for a second, just got to fix this. There we go, glycoproteins. And this, not surprising, is a sugar and a protein stuck together. And we see this on the external surface, the cell membranes. Often they're associated with the mucus and respiratory digestive tracts. And then we have the proteoglycans. Now, this sounds familiar because I just talked about glycoproteins. Now we're talking about proteoglycans. What's the difference? Both of them are a carbohydrate and a protein. It happens to be the ratio and how they're arranged. So in a proteoglycan, I basically have more carbohydrate than protein. So the ratio is different than above. And these are gels, and typically we see these as gels that hold cells and tissues together. They tend to hold a lot of water and moisture within them. You can find them in the umbilical cord, in the eye, and also important with joint lubrication and cartilage texture because of their ability to hold on to moisture and some of their design features, which we'll may talk about down the road. All right, so that was a quick walkthrough of functional groups and carbohydrates. Next video we'll come back and we'll start to look at the world of lipids.